Hi, I'd like to welcome everyone to the PacBio webinar series. My name is Richard Hall and I'm Director of Bioinformatics Applications here at PacBio. Today we're going to focus on variant calling and de novo assembly using PacBio's HiFi reads. First, I'd like to introduce today's speakers. First off, we're going to have Sarah Kingan, who's a staff scientist here at PacBio, followed by Andrew Carroll, who's Genomics Product Lead at GoogleAI. We have a lot of great material to cover today, and the presentation portion of the webinar will be followed by a question and answer session. That's after both talks. You're welcome to submit your questions at any point during the webinar by typing them into the area provided on your attendee dashboard. We will be recording this webinar and making it available for download in the next few days. Just please keep an eye out for a follow-up email with a link to the recording. So with that, we'll get started, and I'd like to welcome Sarah.
Okay, great. Thanks, Richard. And thanks to everybody for attending the webinar. As Richard said, my name is Sarah Kingan. I'm a staff scientist in bioinformatics applications at PacBio, and I'll be covering the de novo assembly with HiFi portion of the webinar. So I'll begin today with an overview of the PacBio HiFi data type. Then I'll show you how HiFi reads can be used for de novo genome assembly, the advantages in terms of the workflow and the computation, and how HiFi data performs in terms of assembly quality, contiguity, and phasing. And then I will conclude with project design recommendations on the new Sequel II system. So first, an intro to HiFi reads. When we think of DNA sequencing technologies, we often think of there being a trade-off between read length and accuracy, with the dominant sequencing technologies providing either very accurate short reads or less accurate long reads. For years, PacBio has offered continuous long read sequencing, or CLR. Continuous long reads have three characteristics that make them an especially powerful technology. Namely, the reads are long, they have a random error profile, so can achieve high consensus accuracy, and lastly, PacBio data shows minimal GC or base composition bias, so you have even coverage of PacBio data across most of the genome. For these reasons, PacBio has been selected as the core technology for many large-scale genome initiatives because it can be used to generate reference quality genomes that are contiguous, complete, and correct. However, recently, in addition to the continuous long read data type, PacBio has developed a new sequence type called HiFi reads, which are both long, between 10 to 20 kb, and accurate, with accuracy of 99.8% on average. This new HiFi data type challenges the idea of a trade-off between length and accuracy. HiFi reads are generated through a process called circular consensus sequencing, or CCS. CCS has always been available from PacBio, dating back to the original 2009 science publication, but innovations in our sequencing chemistry in the last year have enabled much longer high accuracy CCS reads, which we now refer to as HiFi reads. To generate high accuracy reads with CCS, we take advantage of the circular PacBio library molecules. Library molecules are shown in the upper right of this slide, where a double-stranded DNA template is ligated to hairpin SMART Bell adapters. When the sequencing process occurs, the polymerase can make multiple passes around the circular template molecule, generating a long polymerase read. Polymerase reads are then processed into individual subread sequences, which each represent the template, and finally, a consensus sequence of the subreads is generated. And this high accuracy consensus sequence is a HiFi read. Here's a comparison between traditional NGS reads, PacBio CLR, and PacBio HiFi reads in terms of their length and their accuracy. PacBio HiFi reads have accuracy nearly as high as NGS data, with read lengths in the range of 10 to 20 kb. This read type can be used for comprehensive genomic analysis, including de novo genome assembly, which I will discuss shortly, as well as variant detection, both single nucleotide variants as well as larger structural variants, which Andrew will discuss later in the webinar. So now let's talk about the advantages of using HiFi reads for de novo genome assembly. Many of you will be familiar with the de novo assembly workflow for CLR data and have outlined the approach used by genome assemblers like GNU and FALCON. CLR assembly begins with a computationally intensive round of error correction or pre-assembly. This step involves all-by-all alignment of the continuous long reads and consumes most of the compute time for assembly. But with HiFi reads, the error correction is completed prior to assembly using CCS to make the HiFi reads. So the remaining de novo assembly steps include overlap of the HiFi reads and construction of the assembly graph, which produces the contigs. This is followed by contig polishing through HiFi read alignment to the contigs and consensus calling. By starting with HiFi data, we see dramatic reductions in the compute time needed for de novo assembly. For a human assembly using FALCON on 384 cores, the HiFi data requires 14 hours of compute versus 62 hours for a CLR data set, which is an 80% reduction in assembly compute time. But of course, the computation of the HiFi, of the high accuracy HiFi reads is not free, but has been dramatically reduced in the last software release. And future software releases will be able to generate HiFi reads for one 8M smart cell in 3.5 hours on our standard recommended compute system. In addition to the computational savings during assembly with HiFi data, single molecule consensus with CCS is more accurate than multi-molecule consensus, which is what is used for pre-assembly of CLR data. Here's an example for an inbred strain of rice, Oryza sativa indica, strain MH63. This IGV screenshot shows a region of the rice genome with 2 repeats shown in the top track with the navy bars. And I've aligned error-corrected long reads or pre-assembled reads, also referred to as P reads, at the top and HiFi reads at the bottom. You can see errors in the P reads due to erroneous mapping of CLR reads that originate from other copies of these 2 repeat types elsewhere in the genome. In contrast, there are few errors in the HiFi reads shown in the bottom track. Now let's look at how HiFi assemblies perform in terms of contig level base quality, contiguity, and phasing. First, I'll focus on human, beginning with a preprint from Evan Eichler's lab at the University of Washington. In this paper, Volger et al. Directly compared a CLR and a HiFi dataset for a homozygous human cell line CHM13. They had 77-fold coverage of the CLR data and 24-fold coverage of the HiFi data. They found that their assemblies were of similar contiguity, with contig N50 of 29 megabases in both data sets. And they estimated base QV of their assembled and polished contigs by aligning back sequences from the CHM13 sample to their assemblies, and found that the HiFi assembly had higher base QV, QV45 on the Phred scale, compared to QV40.7 for the CLR data. We've collected our own internal data sets for 2 diploid human samples, HD2 and HD5, which are available at the links shown at the bottom middle of the slide. HD2 is a European and HD5 is a Han Chinese individual. The base QV estimates for our assemblies are shown in the box plot in the upper right. For HD2, we have a CLR and a HiFi data set, and for HD5, we just have a HiFi data set. We estimated base QV by aligning our assembled and polished contigs to the human reference and masking out known variants and excluding non-high quality regions using the genome in a bottle truth set. We counted any remaining differences between our contigs and the reference as errors. And we do this in 100 KB windows, so error-free windows will max out at QV51. You can see that both HiFi assemblies have higher base QVs than the CLR assembly, with many windows perfectly matching the reference and having QV51. Now turning to contiguity in human assemblies, the bottom right figure is from the Wenger et al. BioRxiv preprint, which introduced the HiFi data type. It shows a theoretical prediction of NG50 given read length and read accuracy, where each curve represents read accuracy and a proxy for read length is on the x-axis. In the inset table, we can see that the empirical Kingan 50 values for the two HG2 data sets, the CLR and the HiFi, indicate that the HiFi data results in a more contiguous assembly compared to the longer but the lower accuracy CLR data. Moving beyond humans, here's a look at base quality for two samples for which we have ground truth data sets, the indica rice strain I introduced earlier and a Drosophila melanogaster sample. On the left, for rice, we compared a 30 kb size selected library of CLR data to two HiFi libraries size selected at 11 kb and 14 kb. As in human, the base QV shown in the box plot on the left is higher for both of the HiFi based assemblies. To measure this, we aligned our contigs to an independent MH63 reference and called differences as errors. This is a conservative approach because we don't have the sophisticated masking that is available for the human sample, which explains why the base QVs are lower in these samples compared to human. Interestingly, on the bottom in the table, we can see that the BUSCO scores for the CLR and the two HiFi assemblies are all quite similar, with 98% of the plant genes present in single copy. On the right is a data set that we collected in collaboration with J.J. Emerson and Mehul Chakraborty at UC Irvine. We sequenced a female offspring from a cross between two well-characterized Drosophila melanogaster strains, ISO1, which is the reference strain, and A4. Again, we performed assemblies with both CLR and HiFi data. This sample, the results I'm showing here, was from a trio bin sample, and we assessed base quality of the ISO1 haplotype by aligning to the latest flybase reference sequence. The median base QV is about the same for CLR and for HiFi, although there are more windows with high base QV in the HiFi assembly compared to CLR. For the sake of comparison, I've also included the flybase reference aligned against itself on the left, which shows very few errors. And on the right is an Oxford nanopore assembly of ISO1, which is lower quality. BUSCO scores again look good across all four assemblies, despite the much lower base QV estimated for the Oxford nanopore assembly, which indicates that there are limitations of the BUSCO approach for assessing genome quality. Now let's turn to contiguity in the plant and animal genomes. These are summary statistics comparing CLR and HiFi for the Drosophila data, as well as a Pacific bluefin tuna data set, which was collected in collaboration with Barbara Block, Nate Truelove, and Luke Gardner. All assemblies were done with Falcon. For these two assemblies, we can see that the HiFi assemblies are actually more contiguous than the CLR assemblies. For Drosophila, the HiFi Kingan 50 is 5.27 megabases compared to 3.46 for the CLR. And in the bluefin tuna, we're achieving 11.4 megabase Kingan 50 compared to about 4.6 for CLR. However, when we looked at the Falcon assemblies in the rice sample that I've mentioned already, we see that the CLR assemblies are much more contiguous than the HiFi assemblies. Kingan 50 is 11.2 megabases for CLR versus 3.3 for the 14 kb HiFi library and 11.5 or 1.5 for the 11 kb HiFi library. It turns out that the dominant repeat type in rice are 11 to 13 kb gypsy-like LTR retrotransposons, which explains why the 14 kb HiFi library performs so much better than the 11 kb. But we were still disappointed by these results, so we dug in a little bit to the Falcon code and made some changes to expose some parameters specifically for HiFi data sets. So the modifications that we made to Falcon involved the HiFi read overlapping and filtering prior to graph construction and contig generation. We added the ability to ignore indel differences in HiFi read overlaps, and we increased the stringency of overlaps to take advantage of the higher accuracy of the HiFi reads. Shown in the top left is a table from the Wenger et al. BioRxiv preprint showing that homopolymer indels are the predominant error type in HiFi data. Below, I show a histogram of the percent identity in the rice HiFi read overlaps for all differences in the top, indels only in the middle, and mismatches only on the bottom. And so in the bottom panel, you can see that once the indel differences are ignored, dramatically increase the stringency of HiFi overlap identity in order to remove repeat-induced overlaps. So these two changes, ignoring indels and increasing overlap stringency, boosted the Kingan 50 of the HiFi assemblies. On the bottom right, you can see the two new curves illustrating the higher contiguity of the two HiFi assemblies after the Falcon changes. In particular, the 14 KB HiFi library now has a Kingan 50 of nearly 7 megabases. We're continuing to test these changes on additional genomes, and we'll add these features to the distributed Falcon assembly code soon. It's useful to explore how much coverage is needed for your HiFi assembly. So here is a coverage titration experiment we did with HG2, the human data set. We looked at, in terms of fold coverage, we looked at 10, 15, 20, and 50-fold coverage, which can currently be achieved by collecting data from 2, 3, 4, or 10 8M smart cells on the Sequel II system. The top figure on the left shows that Kingan 50 has gains up to 20-fold coverage, whereas in contrast, in the green line shows that phase block N50 continues to improve even at the highest coverage levels, or 50-fold that we looked at. In the bottom left figure, base QV is high at all coverage levels. Note that the y-axis doesn't begin at 0. We're zooming in on, so all of these have pretty high base quality. But in contrast, look at the green line, the phasing accuracy is lower, below 20-fold coverage. So in general, we would recommend coverage of 15 to 20-fold for HiFi for assembly. But if you're interested in phasing a diploid human sample, we would recommend higher coverage. So concluding this section, I wanted to highlight the changes that we've made to the Falcon and FALCON-Unzip code, which is available in Bioconda, with details at the PacBio GitHub pages shown here. We now have native support for HiFi data, as well as faster read tracking and polishing. And we're doing further testing on the indel masking and stringent overlapping for HiFi reads. And we'll push these changes to Bioconda soon. In addition, our developers are working on improving the phasing algorithm for HiFi data. So let me conclude with some project design recommendations. So many of you are probably already familiar with our recently released Sequel II system. Sequel II features an 8-fold higher throughput compared to the Sequel system, which translates into reduced project times and lower costs, while having equivalent performance to the Sequel system. On Sequel II, we're seeing 12 to 16 gigabases of HiFi reads per 8M smart cell. With this type of yield, you can generate a 20-fold HiFi data set for an 800 megabase genome, or a 15-fold HiFi dataset for a 1 gigabase genome. If you're interested in human assembly, as I said before, we would recommend 3 to 4 smart cells to get 15 to 20-fold coverage. Lastly, here's an overall comparison between HiFi and CLR data in terms of throughput and library requirements and various other things. So in terms of yield on Sequel II, for HiFi, you can expect 12 to 16 gigabases of data versus 60 to 100 gigabases of CLR data. HiFi libraries are typically 10 to 20 KB long, so that's how long the reads are, whereas CLR libraries can be much longer. They're almost always longer than 20 KB, but can be more aggressively size selected. Read accuracy is obviously higher for HiFi, on average 99.8%. versus 89% for CLR. HiFi library construction currently requires 10 to 20 micrograms of high molecular weight DNA. So if you're limited with the amount of high molecular weight DNA that you can collect, CLR is a good option for you. And we even have special protocols that require lower DNA inputs. So if you have, as low as 150 nanograms, you can use the low DNA input protocol for CLR. We're recommending coverage of 20-fold for HiFi 60-fold for CLR, assuming you have a diploid sample. And lastly, the genome size you can assemble with one 8M SMRT cell is 800 megabases for HiFi versus 2 gigabases for CLR. And so I wanted to thank my colleagues at PacBio, in particular Zev Cronenberg, who did a lot of work on adapting Falcon to take HiFi data. Aaron Wanger did the investigation of indel mapping and overlap stringency in the rice, and Greg Young collected all of the rice and the Drosophila libraries. And of course, I want to thank our collaborators, JJ Emerson, Mehul Chakraborty, Barbara Block, Nate Truelove, Luke Gardner, who helped us with the collection and analysis of the Drosophila and the tuna data. So with that, I will turn it over to Andrew Carroll from Google, who will be discussing variant calling using HiFi.
All right. Hi, everyone. My name is Andrew Carroll, and I am in Google Health. Hopefully you can see my screen just fine. I'm from Google Health within GoogleAI, and I'm going to be talking about how we retrained a variant calling method we have called DeepVariant for the PacBio HiFi reads. So for the outline of the talk, first I'll be giving a background of DeepVariant, explaining how it works and what it does. I'll talk about some of the initial work in adapting DeepVariant for the publication, which was put out in early 2019 for PacBio HiFi data. I'll talk about what was involved in training a production model, which we released a few months ago, that can fully support PacBio HiFi. And finally, I'll go into some future development efforts that we have. So let me make sure this is, sorry, make sure everything's working okay. Okay, I think everything is. I'm assuming people will bug me if not. All right, so first a background for DeepVariant. So in the process of analyzing sequencing data in the context of a reference genome, it's very common to generate raw reads, map them to a reference genome, and then use a process called variant calling to identify the positions which are statistically significant to differ from that reference genome. And that it's then used downstream for clinical reports or other research applications. And DeepVariant sits into that variant calling bucket. So the analysis of NGS data and variant calling have properties which are partially unknown as well as fully unknown. And I've tried to go through and highlight how well I think certain of these aspects are understood. An important characteristic is that humans impartially understand a variety of these characteristics. And in addition, there are a number of factors which we don't even know that we don't know. So this problem is impartially understood and is impartially resolved. And this is something that's going to be important to keep in mind as we talk through what some of the potential advantages for a deep learning method like DeepVariant provide. So DeepVariant is a deep learning based approach. And to explain what this is relative to prior methods, I've taken what I consider 3 general classes to computationally approaching problems. And in order to define them, I basically Googled the terms and took the first sentence of the first hit, which was, I think, usually Wikipedia. So you can write specific rules which will map from your data to underlying properties you want to deduce. That's the most straightforward and mathematical direct way to do something. And that's something I call statistical inference. So in machine learning, a human has gone through and identified the features which are predictive for classifying a problem well, and has created a framework which allows computers to fit those properties based on what they're seeing in the data. So it's essentially a production application of statistical inference. In deep learning, the main difference is that humans aren't going through and finding the features that are predictive. Instead, the models are set up to look at the data at a relatively raw level and then determine for themselves what features are important for doing well at the task. And DeepVariant falls into this category. And GoogleAI puts a lot of effort into develop frameworks for deep learning-based approaches in general. So these deep learning neural nets consist of layers of neurons that connect to each other. The lower layers of the network tend to learn local information, more granular things. In vision, this is stuff like edge detection. While the upper layers of the network learn more abstract concepts. and finally leading to a classification. So DeepVariant uses this framework and repurposes image classification architecture called Inception v3, and it takes not quite images of a genome browser, but things which are conceptually similar. So this is an ordering of genomic data with a left coordinate that is in the reference and a right coordinate in the reference. It's a composite of multiple channels, bases, qualities, mapping qualities, strands, and so on. So a human couldn't really meaningfully look at this data, but it's able to be processed in the same sort of an architecture. And the output of this is a classification per position of those which are homozygous and match a reference, heterozygous or homozygous to a variant position, as well as a confidence probability. The first proof of concept of DeepVariant was actually developed for the Precision FDA Challenge, which occurred in 2016. And this was really just to see whether it could be done. The team didn't know whether it was possible to train a competitive method using this approach. And the Precision FDA was the first time there was a fully withheld, this HG002 sample, fully withheld truth set that nobody had seen before, but could be assessed. So DeepVariant was submitted, the early version of DeepVariant was submitted to this competition, and it won the award for highest SNP accuracy. And that provided the validation that this is an approach that could work quite well and become the foundation for the subsequent development. So since that time, DeepVariant has been fully open sourced and released in late 2017. You can see that the overall development timeline for it here. Subsequent to its release, we have continued to improve it. We've trained new models which extends its functionality to exomes, PCR positive samples, improvements to speed and cost, and then most recently in the work that I'm talking here about, extensions to the PacBio HiFi data type. Furthermore, DeepVariant was published last year in Nature Biotechnology. So these are the characteristics that we optimize for when we are trying to make sure that DeepVariant is the best method for users. So the core brand of DeepVariant is it's the most accurate germline variant caller in competitions like Precision FDA and in external benchmarks. So we value accuracy as the most important reason to use DeepVariant. It's robust. We try to make sure its accuracy increases on hard data sets. We target speed. So we have benchmarked less than 5 hours on a single machine with acceleration options that exist as well. We aim for about a $3 raw cloud cost on Google Cloud and hopefully comparable on others. It is extensible, so you can retrain this for new data types and new technologies without the need to write new software. Finally, DeepVariant is open source and it uses standard file formats. So it reads from BAM and CRAM files and writes VCFs and GVCFs. So in addition to that Precision FDA competition, DeepVariant has been benchmarked externally by a variety of groups in a couple different publications. I'm showing you one benchmark from DNA Nexus, which shows the number of errors made for 35-fold coverage in Lumina data. You can see that DeepVariant has a substantial error reduction relative to GATK, and also that as we released new versions of DeepVariant, we continue to improve its overall accuracy. I should finally point out that these accuracy numbers are based on the genome in a bottle gold truth set, and they're based on the confident regions. I'll show you some data later on, which also demonstrates how PacBio can even extend beyond these regions effectively. I think there's an important reason for why DeepVariant is accurate, and this has to do with its understanding of why it makes errors. And this goes back to the properties which I mentioned of sequencing being partially and fully unknown. So what I'm showing you here is a plot where the x-axis is the estimated quality score of variant calling method, DeepVariant or GATK haplotype caller. This is its self-reported probability of error. On the y-axis is the empirical error that you can get by calculating on the gold standard. So what you see is DeepVariant is much better calibrated with respect to why, when it says it's not confident in a variant, it's more likely to have an accurate assessment of that. Whereas there are times when GATK says it's extremely confident that its observed empirical quality is much lower. I think one of the reasons for that is that because GATK is written by humans, there are various nonlinear interactions between errors and things that we don't understand, which make it very difficult to encode all of the reasons why a call may or may not be present. And by modeling the problem in its internal way, I think DeepVariant is better able to assess the unknown unknowns and therefore produce better accuracy as well as better overall calibration to that. All right, so the next story I'm going to tell is about retraining DeepVariant for this PacBio HiFi data set. So in late last year, PacBio reached out to Jason Chin to help coordinate collaboration between Google and PacBio and DNA Nexus around the adaptation of DeepVariant. And so I won't go into detail about the circular consensus sequencing technology, which enables HiFi reads, because you've heard quite a bit about this. What I will highlight is that the types of errors that are present in HiFi reads are just different from the types of errors that are present for short reads. So for short reads, you have these mapping complexities, you have coverage variability, which occurs because it's difficult to map short reads uniquely and because the sampling of the genome with respect to GC content is just more non-uniform. And for the PacBio HiFi set, these reads are much more mappable. They're also much more uniform in terms of sequence context and coverage. But they have just an overall different error profile. Specifically, the homopolymers are a bit noisier with respect to indel lengths in the HiFi data. So here, what I'm showing you here is a good example which illustrates these two differences. So this is a region which is a long TA repeat preceded by a stretch of poly A. And it turns out that there is a heterozygous variant here. And this region would be very hard to map. I'm not sure Illumina would have any reads which overlap this region. And PacBio is able to span this completely, but you see this variability in signal with respect to the indel length. So if you can resolve this well, you'll be able to call this variant that Illumina wouldn't have been easily able to call. Okay, so how do we retrain DeepVariant? The way we retrain DeepVariant is by essentially generating examples that look exactly like we would ask the variant caller to classify. So actually here you see, this is what DeepVariant sees. This is this composite image, but actually what it sees is these dimensions of bases and qualities and so forth. So this is an example for a position. You associate with that with a label, and then you are able to feed this into the model, and the model sees batches of this, and it updates its weights to change the behavior of the overall model. So this relies on the labels that we have from NIST Genome in a Bottle, which are foundational to all the work that we're able to do. And finally, when we train, we always use chromosomes 1 through 19. We'll never train on chromosome 20, nor use it for tuning. And this allows us to make independent assessment without overfitting on chromosome 20. We also generally never train on HG002, except the first data set we had was HG002. So for the PacBio case, we are training over HG002, and as a result, I'll show you the benchmarks only for chromosome 20. As we get more genomes, we're going to move this to the same strategy for the Illumina models where we train on HG001 and 5 and leave out 2, so we can get a full genome assessment of accuracy. So the SNP accuracy, when you retrain DeepVariant for HiFi data, is somewhat improved. Here's the accuracy that you see by just applying the Illumina model from DeepVariant and GATK4 with some filters. So in all these cases, this accuracy is quite high. So 99.5% accuracy for GATK is pretty good accuracy. With retraining DeepVariant, we can get this up to about 99.9% genotype level accuracy, which is quite a high level of accuracy. But for the indels, there are just substantial differences. So GATK here is only able to achieve about 80% accuracy. And the DeepVariant Illumina model probably thinks that these reads look just unusual, and so it is reluctant to call variants here. So that's just because it hasn't seen this PacBio HiFi data before. When we retrain DeepVariant by showing these examples, we're able to achieve an indel accuracy of about 95%, which is pretty high indel accuracy. It's higher than many of the preparations that you would get from Illumina, which involve PCR and use GATK. So this is already looking quite good. There's an attractive feature to this HiFi data, which is when we do coverage titrations, we find that this is actually quite robust to coverage. So you can go down to 10-fold coverage for SNP accuracy, and you can still achieve quite high accuracy. I think one of the things that's driving this is the fact that coverage is more uniform with respect to sequence contexts. And so as you downsample, you don't lose coverage entirely for certain more difficult regions. For indels, you do see a slightly higher dependency on coverage, but you still see this hold up pretty well down to 15-fold coverage will still get you 90% accuracy for the publication level model. And so based on these recommendations, you can choose for yourself how to tune between the number of flow cells that you want to run, smart cells you want to run in order to get 10-fold coverage, 15 or 20-fold based on these titration curves. Okay, so that was the initial take that we took at retraining a model. It involved actually very few code changes and mostly just showing data. From that point, we wanted to see if there were additional ways that we could improve. So one of the methods that we have is the fact that the HC002 genome is in a trio with known variants. So it's possible to phase these reads. So we used a method, what's-hap, actually PacBio used a method, what's-hap, to phase the reads that are present. And then we ordered the reads in DeepVariant based on their haplotype tag, meaning we put all of one parental set at the top and then the other parental set at the bottom. And we trained models so that it could learn now that the local information tends to come from a haplotype. So when we do this, we see a substantial improvement in the overall indel accuracy here. So we're able to go from 95% to about 97.5% indel accuracy. And I'll point out that this is not by providing any additional channels, it's just through reordering information. So DeepVariant is essentially able to now learn that locality has meaning when data is presented in this type. Unfortunately, when we're running in the wild, we don't have access necessarily to haplotype tagged reads. So we're not able to have this particular benefit, but we're aware that this is something that we want to bring in to further improve the accuracy of DeepVariant. Okay. So at the end of this process, we had a model which was able to produce variant calls for publication, and the reception was overall quite positive, both by PacBio as well as by the general community. At that point, we began working with PacBio to see how is it possible to train a better production model and make this generally available to the PacBio user base. So when we're doing this, we have slightly different considerations than for a research model. We want to make sure that we can address all the different needs that the PacBio user base will have. And that might be to run the 1M chips or the 8M chips, to sequence at various depths that balance cost and accuracy, to use different insert sizes to have potentially longer HiFi reads or HiFi reads that have more passes, to handle the variability across instruments or library preps. We also wanted to be robust to the high coverage sequencing of amplicons. So the challenge we had is, can we train one model that will be robust to all these use cases, or we need to train many possible models? Working with PacBio, thanks to some of the data they provided, we trained models that have a mix of different chip types, of different insert sizes, and of different coverages, spanning from 9-fold coverage to 30-fold coverage, and then we had one with 82-fold coverage to be able to cover that high amplicon case. So this created many hundreds of millions of training examples across 27 different genomes. So this resulted in a release model, version 0.8, which we put out in April. So we have a case study, which will walk through exactly how you apply this to the HiFi data. It will handle a 30-fold genome in about 5 hours on a 64-core machine. So that's about 320 CPU hours, which is similar to the amount of time it takes for Illumina genomes as well. So this is a pretty fast overall compute time. And so this is a fully supported data type. We'll be continuing to update it and improve it. And going forward, we'll basically fully support the PacBio HiFi reads here. So when we assess this release model, and this is actually something we worked towards, it substantially outperforms the publication model, in part because it has more data. One of the things that we found is that the mixed model on the 1M and 8M chips and with various insert sizes outperforms a model that's trained only on 1M data, and it outperforms a model that's trained only on 8M data, and each on their own data type. It is also robust across various insert sizes. So one of the reasons for this is deep learning methods often benefit from more data, as well as more difficult and varied data. So this forces the models to learn more generalizable concepts. I think this is responsible for why the release model is substantially better than the production model. I'll also point out that this still doesn't use the haplotype sorted tags. So we think there is some additional gain that we have to make on indel accuracy. And one of the reasons I'm showing you indel accuracy is that the SNP accuracy is very similar in all these models. I think that in each case, the accuracy of this model probably exceeds the accuracy of the truth set. So the SNP variants are in pretty good shape in each of them. Finally, we look at the high coverage model. So with 82-fold coverage, this is able to be quite robust and capture the additional information. So we can get to 99.4% indel accuracy on the high coverage 82-fold model. So actually this is the release model on the 82-fold coverage data. So if you're doing amplicon sequencing, we think that this will just continue to increase in accuracy up to 100-fold coverage or more. Okay, so what I've shown you so far have been benchmarks on the confident regions. We took some assessment as to whether PacBio is improving calls in the non-confident regions. So as you may know, the genome in a bottle assesses each of these regions for callability. There's a set of regions which are determined to be difficult or non-callable. So what I've done here is I've taken the Q20 DeepVariant calls that are outside the confident region. So this is the ones where DeepVariant is relatively confident that this is a variant position. And as you can see, the TI-TV ratios, which are an indirect measure of quality, look pretty similar between PacBio and Illumina data, but you're able to make more of these calls on chromosome 20. So you have like a little more than 1,000, so about 10% more calls that are made with high confidence outside the confident regions on chromosome 20. And so if you extrapolate from the fact that chromosome 20 is about 2% of the genome, there would be many tens of thousands of additional high confidence calls that you could make with this data type. We're eagerly awaiting for an extension of the confident regions, which we think this data type will allow Genome in a Bottle to conduct, and I think that's going to improve things even further. So in terms of future development efforts, just a few things to highlight here. Oh, sorry. One is that we had an unexpected finding. We identified that DeepVariant is calling structural variants in this HiFi data, even though it was never trained to do so. And in fact, because the truth set is a small variant truth set, anytime DeepVariant would see these examples and it would see them in training, it would always be penalized for the call. So it's quite interesting that this behavior was able to be retained even. It's shocking. So we're trying to investigate exactly how we can extend this functionality. Basically, I think what this means is that there are some generalized concepts which are important for indel calling, that it's more expensive for DeepVariant to unlearn and gain the accuracy of not calling structural variants on that truth set than it is to retain. So that's an interesting observation. So when we asked Aaron from PacBio to assess this, it turns out these calls are made with quite high precision, but they're made with generally low recall. The deletion recall is a function of the fact that we actually don't show PacBio windows longer than 221 base pairs. So it actually can't see any event, so it won't call something longer than 221 base pairs for deletion. But the insertion, the way we expose the data, actually shows the insert, so it can actually recover quite a bit of these insert calls. So when the calls, if you run DeepVariant, you'll see long events. It won't capture all your structural variants. We still recommend you to run a dedicated structural variant caller, but the events that are found are at generally high precision. So the future development efforts, we want to continue to improve by identifying more and diverse data sets. We think this is going to continue to make DeepVariant better for HiFi data. We're eagerly anticipating extensions to the Genome in a Bottle truth set. By extending it, we think that this will allow us to train and perform even better on the non-confident regions, and hopefully on the confident regions as well. We're trying to find ways that incorporate that haplotype phasing information to gain that additional performance boost. We're looking at improved representations. So we're basically trying to find if we can make DeepVariant understand the structural variant data type, and therefore do much better about calling that to enhance its ability to call variants across all sizes. We continue to improve the speed and cost of DeepVariant. So we've driven that down with every release. We've driven the cost down, speed up at every release, and we plan to continue to do so. And finally, we're working on some exciting new genomic methods that hopefully I'll have a chance to talk about at some later time. Finally, I want to thank our external collaborators, in particular Jason Chin, who initiated this collaboration, as well as Kieran from the Broad, Fritz from Baylor, Shilpa from Harvard, as well as Aaron and Billy, who we work closely with at Pacific Biosciences. Finally, we have a team blog. It's at our GitHub account, where we do various investigations. You can read some of our prior blogs. We have one on the PacBio data, which goes into detail and presents some of these numbers as well. And we'll be continuing to put out new and interesting analyses there. Finally, this is the genomics team in Google Health. I want to highlight a couple people who contributed to this work. So this is Pichuan Cheng. She is the tech lead for DeepVariant and generally oversees the engineering effort for DeepVariant. Alexei Kolesnikov trained both production and release models. And Sid and Gunjan are working on some of the exciting extensions that hopefully I'll be able to tell you about in the near future. So that concludes my talk. Thank you very much for taking the time to listen. I'll be happy to answer the questions that you have.
Hi, so this is Richard again. I'd like to thank Sarah and Andrew for those great talks. We do have plenty of time for some questions. So to start off, for Sarah, how do HiFi reads, assemblies, and CLR reads with equal N50 compare? Aren't you comparing assemblies made with reads of different N50s? Have you tried taking the same tissue and using one with CLR and one with HiFi and comparing the assemblies?
Yeah, that's a good question. So the first one was the read lengths are different for the HiFi versus CLR. And what if we constrain the data to have the same read length but different qualities? And we've done that experiment. We've taken CLR reads and truncated them to be the length equivalent to a HiFi library, and the assembly performance is pretty poor. So having the higher accuracy is a benefit for sure. The other question was, oh yeah, the same tissue and sending it to two groups, one with CLR and one with HiFi. That's actually what we did with the Drosophila data set, the rice data, and the HG2 human. So for all three of those samples, we started with the exact same genomic DNA, and we just made HiFi libraries and CLR libraries from the same starting material. And generally, you know, the top line summary is base QV is higher for HiFi. The contiguity kind of differs depending on the organism. So, some with rice, you know, the contiguity is higher for CLR, but with Drosophila and rice, the contiguity was higher for the HiFi. And yeah, so we kind of did that in the experiment. I talked about it in the talk.
Thank you. And another one for Sarah. How does a low input DNA CLR assembly compare to a normal input DNA CLR assembly? Okay,
so the low DNA input protocol works by removing the size selection step during library production, just to prevent loss of DNA. And so the result of that is that the read lengths tend to be shorter for low DNA input libraries, and the yields tend to be lower as well. So it's always better to do size selection if you can. So the low DNA input protocol is mostly designed for samples that cannot be used on standard CLR libraries. So if you're able to do a standard size selected CLR library, you should definitely do that because it gives you better yields, longer reads, and better assemblies.
Okay, thank you. And sorry to jump around, but just so that we have time to get both people answering questions, I'd like to jump over to Andrew now. So will DeepVariant be trained for long-read RNA-seq data, both PacBio and ONT, including cDNA and direct RNA-seq on ONT?
Yeah, this is a good question. So let's break down the two different components to this. So one of which is, can we train DeepVariant to call variants in RNA-seq data in some form? So this is a project that's actually been of quite a good deal of interest. It's been on our radar to conduct. We think it's relatively easy to do with respect to the generation of the data that would allow us to train. So yes, we want to do that. It will be possible to do that for either PacBio data or for Illumina data. We just need those data types. So I think the question of, I think this will be done. I think the question of timing has to do with when there's a good data set that will enable us to do that. And so far we've been hoping that partners will come to us with that. With respect to the various data types, so DeepVariant can be trained for Illumina, PacBio, or Oxford. We have to, in order to operate for noisy data, either the CLR reads or Oxford Nanopore, we actually have to do some changes to the candidate generation logic. And that actually requires us to write some interesting software and heuristics and algorithms. So that is not as straightforward an operation to do as it is to plug into our existing training infrastructure. So although that's possible, we're not sure how to prioritize that work. So it may be done, but I couldn't commit to the long, noisy read. For the reliable isoseq data, it should be doable with the right data set.
Okay, thank you. So I'm just going to group a couple of questions here on kind of the more practical side. But is there a Conda package for DeepVariant for Mac? And with the current Conda release, is that the latest CCS HiFi model included with that release?
Yeah, so we distribute DeepVariant as a Docker image and as a singularity image. And a couple members of the community have adapted this into Conda installable packages in some form, but I haven't personally gone through the installation experience. So I think it's likely that one of these Conda packages can work. And if you have questions about this, you can file an issue on our GitHub. It's watched by our full team. It's also watched by some of the community members who have generated these Conda images. Actually, now that I think about it, I think Billy Rowell, who's in PacBio on the call, may have contributed to some of this. So this would be one of the installable ways, and it should, these images should contain the most recent v0.8, which includes the PacBio model. Basically, if you go to our GitHub and look at the quick start for PacBio, you'll see that you can put in for model type PacBio. If that works validly, then you've got the right version.
Okay, thank you. And just going back to Sarah, so for phased genome assembly, could CLR be used for the primary assembly and HiFi used to replace Illumina in the polishing step to correct homopolymer indel issues in the same way that short reads are used?
Sure. So there's kind of two common sources of indel errors in polished assemblies. The first is the homopolymer indel issue, which exists for both HiFi and for CLR data. But with diploid assemblies, sometimes we observe indel errors in polished contigs that originate from pooling reads from the two different haplotypes. So if you're able to bin your reads by haplotype, either with like the trio binning approach developed in Adam Phillippe's lab, then the polished contig QVs tend to be the same for CLR versus HiFi, and I showed that with the Drosophila data. We trio binned that sample, and the median base QV for HiFi and CLR assemblies was the same. So if you're able to phase your reads up front, then that kind of gets rid of that common mixing haplotypes based indel error. And in general, we're starting to shy away from polishing with Illumina reads because sometimes that can kind of erase haplotype differences or differences between repeat copies that exist in the genome. And so it can have can do more harm than good to polish with Illumina data.
Thank you. And for Sarah, is there a pipeline for assembling HiFi data that's currently available?
Yeah, so we've adapted the Falcon code to support HiFi data, so you can use that, and that works right out of the box. It takes FASTA inputs into Falcon, and we're going to be exposing more parameters soon that are useful for HiFi assemblies. You can also use other assemblers like Watchtower, WTD BG2, works with HiFi data. There's a CCS mode in the command line for Watchtower. And I know some people have been using Fly to assemble HiFi data. I haven't tested it myself. Yeah, so the main assemblers that I would recommend for HiFi are Falcon and Watchtower.
Okay, thank you. And just jumping back to Andrew now, because I know he does have a hard stop at 10 a.m. So was there any specific reason to choose chromosome 20 for DeepVariant training?
Yeah, that's a good question. So actually, to be clear, chromosome 20 is withheld from training. So it's used as the independent evaluation set. The only reason, so basically we wanted to avoid chromosomes which had a genome structure that was a little bit unusual. So we exclude chromosome 6 because we don't want to capture the MHC regions. We exclude chromosome 19 because its genome structure is a little bit different. We just wanted something that was representative and which would allow us to use most of the genome for training. So, 98% train and then 2% for the independent eval, given that we also have a couple full genomes that are held out generally, seemed like a pretty good mix for that. As I said, the more training data, the better. So, keeping the larger chromosomes for training is quite useful.
Okay, and again, for Andrew, any plans to enable DeepVariant to run on GPUs?
It's great. This is a great question. I removed the slides on acceleration. So DeepVariant does work on GPUs, actually runs quite a bit faster. For what our team has produced, the call variant stages accelerates quite a bit on GPUs. There is a company called Parabricks, which has done a full GPU acceleration end for end, so including the initial candidate generation as well. And finally, Google has these instances called TPUs, which are specifically designed to process TensorFlow code quickly. So DeepVariant will also accelerate via TPU. You can, in our case study, you should be able to see examples for CPU and GPU based running, and it will go quite a bit faster with those GPUs. The benchmark numbers I presented were CPUs.
Okay, thank you. And I know Andrew does have to leave in a minute, so I'm just going to group some of these questions together and kind of make my own interpretation. But broadly, what are the prospects for using DeepVariant on unusual organisms and organisms that are more than diploid, and for cases where we have population studies?
Yeah, so this is a very good question. So we're starting to get into this. We conducted an investigation last year where we looked at mosquito genomes, and we found that DeepVariant performs much better when we retrain based on pedigrees in the mosquito genome. So the human genome structure looks different from the genome structure of other organisms. That's with respect to our genetic diversity, which is much poorer in humans, as well as with respect to potential differences in ploidy and segmental duplications. So we're exploring the ability to train plant models or non-human models that should give tailored performance to more complex genomes. I will point out that we've done a benchmark on the 3,000 rice genomes, so we have some metrics which indicate that it performs quite well. But I think in the near future, we'll be looking to put out a non-human model. And if you have, if there are people on this call who have data which they think is possible to construct a truth set with that, it would be interesting. You can reach out to me. We can see if we can put together a model for that organism.
Okay, thank you, Andrew, and thank you for your time today. I know you did have a hard stop at 10 a.m., but we do have some more time for Sarah to answer a couple more questions. So given that we still have some attendees, I'll go back to Sarah with some questions. So for the rice example, it highlights the effects of different repeat composition on the final assembly. For genomes with high repeat transposon content, would you recommend using HiFi reads or CLR?
Yeah, we haven't really done the kind of head-to-head comparisons for HiFi versus CLR for any like really big complex genomes. I think human is the biggest that we've done. When you get much bigger than 2 gigabases, the number of cells required to generate enough coverage for assembly tends to diverge. If you're under a gig, then it's 1 cell HiFi, 1 cell CLR. For human, you're looking at 2 cells of CLR or 3 or 4 cells of HiFi. So there are obviously some cost considerations there. You know, and I think we're making a lot of progress on HiFi assembly, kind of reparameterizing it, reparameterizing Falcon to not collapse repeat copies of the genome. So, I mean, right now the CLR solution works really well if you can get like a very aggressively size selected library for a large repeat-rich genome, like that's going to work. But I'm optimistic that, you know, in the next 6 months or so, HiFi is going to be performing a lot better in those types of complex genomes as well.
Okay, thank you. So we'll have one last question for Sarah before we wrap up. So how easy is it to prepare HiFi libraries compared to a CLR prep?
We're still, I think we have a protocol that's released. Maybe when we release the webinar slides, we can include a link to the protocol. I haven't done it myself because I'm a bioinformatics scientist. And I think it takes, I don't know, Richard, if you have, you might know more about this than I do. I think it takes like about a day and a half to do the prep. Yeah, Richard, do you have any?
As it currently stands, the prep is a little bit longer, but it is in active development. And I think very soon the prep will be in line with CLR. It isn't significantly more difficult. There is a higher requirement for DNA. I mean, we've seen that the low input DNA prep is really only for CLR. But other than that, it isn't an onerous prep to get that kind of DNA, because the big difference is for a HiFi prep, we actually don't need really long fragments of DNA. So you don't have to be as careful as you would if you were running a 30 KB size selected library trying to get 100 KB fragments, which obviously are really prone to being fragmented during preparation. Mm-hmm. So for some things it's easier, and for other things it's harder, but we're currently working on high throughput, faster prep for HiFi libraries. Okay, so again, I'd like to thank Andrew and Sarah for their time today. This talks were really great. And just in closing, I want to briefly mention a couple of things. So we have recorded this webinar and it will be available for download in the next few days. So please just keep an eye out for a follow-up email with a link to the recording. Secondly, you can stay up to date on up and coming webinars, as well as conferences and trade shows you can meet us at by visiting and bookmarking pacb.com/events. And if you have an interesting research project that could benefit from smart sequencing, consider applying for one of our smart grants and win free sequencing. For more information on the different smart grants that are currently ongoing, again, you can visit pacb.com/smartgrant, SMRT-GRANT. So again, thank you all for your time today. I hope you've gotten something out of this webinar, and we hope to have you join us again in the future for one of our up and coming webinars. Thank you very much and have a nice day.